In this first episode of Toxicity Tests, we're going to look at acute systemic toxicity. We will be looking at 10 or so slides from the AcuteTox website, and then we will look at 7 or 8 slides from the AcuteTox project, which was an EU project. So this is the first slide I'm going to use from the AcuteTox website, and it gives us a definition of acute systemic toxicity. Acute systemic toxicity testing is the estimation of the human hazard potential of a substance by determining its systemic toxicity in a test system, currently animals, following an acute exposure. Its assessment has traditionally been based on the median lethal dose, LD50 value for short, which is an estimate of the dose of a test substance that kills 50% of the test animals. For a substance to have a systemic toxic effect, it must be absorbed by the body and distributed by the circulation to sites in the body where it exerts toxic effects. So that's just a broad definition of acute systemic toxicity. This next slide gives a little bit more detail about acute systemic toxicity testing. It reads, conventional animal tests for acute systemic toxicity are rooted in the 1920s concept of the median lethal dose, or LD50, which is the single dose of a test substance that can be expected to cause death in 50% of the animals in the experimental group, as we've just heard. It goes on, since their conception, LD50 type studies have become the most pervasive in vivo tests in most regulatory toxicology frameworks, and are often conducted via more than one exposure route, oral, inhalation and dermal being the most common, but also intravenous, intraperitoneal, which is into the abdomen, and intramuscular are also performed. It has been postulated that the regulatory appeal of LD50 type studies may lie in their substitution of a simplistic index of toxicity in place of real-world biological complexity. This slide describes the OEC test guidelines for acute systemic toxicity testing. The test guidelines are 402, 403, 420, 423 and 425. There was one called 401 for acute oral toxicity, but this was deleted in December 2002 because it was considered too inhumane. Anyway, 3 lines down it reads, 3 reduction alternatives to the oral LD50 test have been developed and validated. These are what vivisectors call refinement tests, one of the 3 R's. Anyway, it says the fixed dose procedure, OECD TG 420, the acute toxic class method, OECD TG 423, and the up and down procedure, OECD TG 425, are examples of this refinement method. Each of these methods uses preset doses with the starting dose based on a small range finding study, cytotoxicity screens or pre-existing data. Following dosing, typically by gavage, which is a tube down into the stomach, the animals are monitored for overt toxicological signs until death, and that's under the acute toxic class or up and down method, or evident toxicity in the fixed dose procedure. So obviously the fixed dose procedure is the most humane out of those 3, because the other 2, although they're the refinement methods, they sound pretty awful if overt toxicological signs are written down or noted until the death of the animal. So it doesn't seem to be that much of an improvement. And it should also be noted that acute inhalation toxicity test still takes place, and acute dermal tests still take place as well. So they've only got rid of the acute oral toxicity test at the moment. So these are some of the worst tests for the animals, as perhaps we will see later on. This slide concerns itself with barriers to progress and what could be possible if those barriers were overcome. It talks about the NRC 21st Century Vision Report, which was commissioned in 2007 by the US Environmental Protection Agency. It says, current toxicity testing practices are long established and deeply ingrained in some sectors. Thus, some resistance to the vision proposed by this committee is expected. This may partially account for the underwhelming rate of progress in some parts of the globe, despite the near consensus view expressed at the October 2000 International Workshop on in vitro methods for assessing acute systemic toxicity. The near consensus view that was expressed said that if there was a commitment to conducting a formal validation study, and this commitment was strong enough, along with scientific resources that could be harnessed for this effort, and if the result was that the in vitro test studied proved good enough, a replacement test battery might be achieved in as short a time as 2 or 3 years. So it seems that it's got less to do with the impossibility of replacing the animal tests and more to do with long-standing traditions and the unwillingness to change. This slide is about whether the LD50 test is actually scientific or not, and it also points out the fact that the LD50 tests have never been formally validated. It reads, despite their continued presence in international toxicological testing frameworks, the LD50 and related methodologies have been among the most widely and harshly criticized on both scientific and ethical grounds. It has been stated, for instance, that for the recognition of symptomatology of acute poisoning in man, and for the determination of the human lethal dose, the LD50 in animals is of very little value, and even if the LD50 could be measured exactly and reproducibly, the knowledge of its precise numerical value would barely be of practical importance, because an extrapolation from the experimental animals to man is hardly possible. So they seem to be saying there that even the LD50 value is not really known in animals because it's not reproducible every time and the value is not agreed on every time. I must admit, I did think they did actually know what the LD50 value was in animals, but if it's not reproducible, it's not science, I'm afraid. Anyway, it goes on, it has likewise been pointed out that the test has never been formally validated, like I said earlier on, and the widespread use of the test has therefore not been based on a documented good performance, but rather on the lack of better tests. This slide expresses how uninterested the pharmaceutical companies are in knowing the LD50 value of a drug. It reads, on the basis of an extensive intercompany data sharing exercise, the expert group determined that the information obtained from conventional acute toxicity studies is of little or no value in the pharmaceutical development process. The conclusions of the expert group were subsequently considered and endorsed by regulators and scientists from the EU, the US and Japan at a workshop in November 2006. Results from the quantitative surveys completed at the end of the workshop further revealed the following. 100% of respondents found data from acute toxicity studies of little or no use and only used the information in dose setting for other studies in exceptional circumstances. 100% of respondents agreed that they would not carry out acute toxicity testing if it were not a regulatory requirement. 100% of respondents agreed that acute toxicity studies were not used to identify target organs. 100% of respondents never use acute toxicity data to help set the starting dose in man. 81% of respondents thought the data obtained from acute toxicity studies was of no use to regulators or clinicians. So the impetus for undertaking the acute systemic toxicological studies is not coming from the pharmaceutical industry. In fact, the pharmaceutical industry seems to be totally happy with the idea that they wouldn't have to do these tests. This last slide leads us neatly into looking at the AcuteTox project mentioned at the beginning of this piece. It reads, "Whatever the ultimate reason for the delay, it is simply unacceptable that in the 21st century the state of the art for acute systemic toxicity testing should remain tethered to an 80-year-old, inadequately validated methodology that subjects animals to extreme, unrelieved suffering to the point of moribundity, which is near death, or death. There is urgent need for high-level commitment from member countries of the Organisation for Economic Cooperation and Development to buy into the findings of AcuteTox and similar initiatives, and to participate actively and constructively in follow-up R&D, validation and peer review activities as needed." So here we are at the first slide from AcuteTox. It says a short description of the AcuteTox project. The AcuteTox project has set the very ambitious overall objective of developing an in vitro test strategy sufficiently robust and powerful to completely replace in vivo testing of acute toxicity of chemicals. And under AcuteTox facts, it said it ran from 2005 to 2010. It had 35 partners from 13 European countries. It had a €9 million European grant from the EU, and its overall budget was €15 million. This second slide from the AcuteTox newsletter from April 2010 gives a little bit more detail about the project. It reads, "AcuteTox is an integrated project funded by the EU with the aim to develop and pre-validate a simple and robust testing strategy based on in vitro and computer models for the prediction of human acute oral systemic toxicity and classification of chemicals into the different EU and GHS toxicity classes. Fast, cheaper and scientifically better tests for testing chemicals, cosmetics and other products are urgently needed to replace the animal acute oral toxicity test used today for regulatory purposes." GHS stands for Global Harmonized System and is a UN initiative. This slide shows the Globalized Harmonized System toxicity classes. It reads, "The globally harmonized system is an internationally standardized system for classifying chemicals. For acute oral toxicity, the globalized harmonized system categories are assigned based on the LD50 values, i.e. from animal tests. GHS1 is an LD50 less than 5 milligrams, fatal if swallowed. I imagine cyanide would go into that category. The next one is GHS2, LD50 greater than 5 milligrams, but less than 50 milligrams, fatal if swallowed. GHS3 is LD50 greater than 50 milligrams, but less than 300 milligrams, toxic if swallowed. GHS4, LD50 greater than 300 milligrams, but less than 2000 milligrams, is harmful if swallowed, if a chemical is in that category. And finally, GHS5 is an LD50 greater than 2000 milligrams, may be harmful if swallowed, or it's an LD50 value greater than 5000 milligrams, and this is not classified. So you can see the impetus to know or wanting to know the acute oral toxicity values comes from the globally harmonised system, which, like I said, is a UN initiative, and the EUCLP system also wants to know the acute oral toxicity values because it needs them for warning labels to put on bottles and other packaging. If you are a scientist given the task of assessing the goals of the AcuteTox project, one of the first things that you will need to do is generate a known, reliable data set of poison information, i.e. human acute toxicity levels, which you can then compare the in vitro and in silico test results against. Obviously, you will then be able to judge whether the in vitro and in silico tests that you have selected are predictive or not. This slide describes how AcuteTox has tried to generate such a data set. It reads, "The in vivo data from both animal tests and human poisoning accidents have encompassed 100 to 140 chemicals and have primarily been used in evaluating and calibrating in vitro data and the in vitro test strategy. However, the animal tests currently used to assess acute toxicity for regulatory purposes have not been properly validated." Because the animal tests haven't been properly validated, the AcuteTox team were forced to do retrospective biometric studies. So it goes on, retrospective biometric studies have therefore been carried out within the AcuteTox project to evaluate the quality and predictive capabilities of the in vivo data and have thus provided a reference as to how accurate the in vitro test strategy has to be to achieve regulatory approval. Now, the scientists I'm largely including in this quite long piece on toxicity testing question the scientific validity of this exercise. They maintain that any inclusion of animal data will only confuse and mislead toxicologists. They therefore suggest that any database that you are going to compare your experimental results with should only ever be compared to known in vivo human data, such as you can see on the right hand side of this table. They would say don't include the data from the left hand side of the table, i.e. the animal LD50 test data, because it will only ruin the purity of your eventual conclusions. This slide shows AcuteTox's best performing in vitro assays, which were selected as candidates for the final tiered testing strategy. Sadly, AcuteTox evidently compared their selected in vitro and in silico tests against both animal and human data, making a clear definitive result almost impossible. The second half of this slide reads, "The largest section of the database is of the part containing the in vitro data from all assays evaluated in the project. It also contains in vivo LD50 data on the 97 project chemicals, 50% of which were drugs and 30% industrial chemicals and 20% others, as well as human data, that's human lethal blood concentrations." And it ends, "All in vivo data were collected from the literature." Obviously they probably mean LD50 tests from animals when they say in vivo data, because they nearly always do. However, we mustn't forget that you can also get human in vivo data. So what were the main achievements of the AcuteTox project? Well, let's remind ourselves what the project was about. This slide reads, "The AcuteTox project undertook for the first time a challenging goal to create an integrated testing strategy to replace the animal testing used today for predicting human acute oral systemic toxicity. And this AcuteTox project was based exclusively on in vitro and in silico methods." The achievements are summarized in these 4 paragraphs. They read, "An in-depth statistical analysis of the large data set generated in this project resulted in a list of 8 in vitro and in silico methods, which resulted to be the most promising for inclusion in proposal of potential testing strategies. Protocols of all these methods will be available to the public as INVITTOX protocols through the ECVAM database on alternative methods." The second paragraph reads, the last phase of the AcuteTox project focused mainly on the assessment of the predictive capacity of the proposed tiered testing strategies and the identification of assay combinations that give the best prediction in terms of classifying chemicals into the official acute oral toxicity categories, GHS and EU system. 5 proposals for the in vitro tiered testing strategies were formulated and evaluated in terms of predictivity. The third paragraph reads, the outcome of this analysis reinforced previous results obtained with the 3T3NRU assays and supports the use of this validated cytotoxicity assay to identify unclassified substances LD50 greater than 2000 milligrams as a first step in a tiered testing strategy. The final paragraph reads, several in vitro assays have proved to be useful to identify alerts for tissue-specific toxicities such as neurotoxicity and nephrotoxicity. However, the results of the classification analysis showed that complementing the 3T3NRU assay with those in vitro assays is not improving significantly the classification of compounds in toxicity categories 1 to 4. This outcome is largely linked to the fact that the current classification systems are based on arbitrarily assigned cutoffs for the rat LD50 values and do not include more detailed scientific mechanistic information on the compounds. Thus, a revision of the current classification schemes might be advisable and should be put forward to the European regulatory agencies. The AcuteTox project echoes these sentiments under the recommendations that it makes. The first paragraph reads, the results obtained from the classification analysis performed in the AcuteTox project lead us to question the scientific motivation for the current classification systems for acute oral toxicity and to suggest the revision of the globalized harmonized system and the EU CLP systems. I do have some sympathy for the AcuteTox project trying to validate their test results with LD50 test results from rat studies. Anyway, this second paragraph, which will be the last paragraph I'm going to read out, further reiterates this particular point and the confusion that it can cause. It reads, the outcome of the analysis for consistency in classification GHS and EU categories corresponding to reported ranges of LD50 respective of individual 97 substances included in the AcuteTox project give an indication of potential consequence for ambiguity in corresponding classification. The analysis showed that with at least 90% probability, 50% of the substances would be unequivocally classified by a single category. 40% would ambiguously occur within the limits of 2 adjacent classification categories, and 10% of the substances have LD50 ranges of sufficient scope to span 3 or more different classifications. This analysis reinforced previous findings by Rudin and Hansen and leads to the same recommendation of revision of the GHS-CLP system.